Hey guys, how are you doing? Today in this video we are going to talk about cell biology and particularly about prokaryotes versus eukaryotes. Biology has become full-fledged science for last 70 plus years, typically with the discovery of double helix structure of DNA, and this was discovered in 1953 by Watson and Crick. So full-fledged science means we have some basic laws or principles which can be applied to some interesting biological problems. For example, how does our immune system function? How does modern medicine functions? How does nervous system function? So on and so forth. And about cancer, basically, typically about cancer, right? So let's begin our discussion about cell. What is cell? Cell is a structural and functional unit of life. Okay. So let me use my pen, paper. And here we go. You have done this in class 9th and this is very important for your PT examination. In previous years you have seen many questions about cell, right? What is cell? Cell is by definition functional and structural, or you can write first structure then function, right? So it is a structural and functional unit of life. It gives you some kind of structure, a structural and functional unit of life. Functional unit of life. For example, if you have to build a house, what do you need? You need some bricks, you need some basic building materials. Similarly, if you want to have life, you have to have a cell, So cell is typically something which you can call a building block of life, right? So it is what? It is a building block of life, right? It has everything, whatever an organism needs. So if we were to talk about, organism can be of two types. One are unicellular. Unicellular organism has one cell only, and there are multicellular organism. Multicellular organism like us, unicellular. For example, we have unicellular bacteria, and typically bacteria. You have yeast also, which is unicellular, right? You have, for example, protozoa, right? They are unicellular protozoa. For example, amoeba and paramecium, they are unicellular animals. Zoa means animals in science terminology, paramecium. Where you have read this, all of these things, you have done this in your 9th class, right? Okay, I hope sabko yaad bhi hoga, right? So if we were to talk about evolution of Earth, Earth was born around 4.5 billion years ago, right? So there are some red, so there are some red letters dates. For example, 4.5 billion years ago, Earth was born. This was the event where Earth was born. And after, 0.5 billion years later, around 4.0 billion years ago, Earth was cooled down. Earth was super hot initially, right? Earth was cooled down so that it could, harbor life, right? So there was possibility of life. And what happens next? Around 3.7 billion years ago, first life was formed. 3.7 billion years ago from now, first life evolved on the Earth. First life evolved on the Earth, right? And those were basically bacteria. So this kind of life was unicellular life. They were very primitive type of cells, right? They are very primitive type of cells. And from here we can distinguish, and let me tell you another date. And for a long gap of 2 billion years, can you imagine, there was no evolution. There was only, nothing, 2 billion years. After 2 billion years, gap of 2 billion years, around 1.5 billion years ago, first well-developed, highly organized cell was evolved. Organized cell was evolved, right? And that cell, for example, in your screen, this cell, this is called eukaryotic cell. This cell is called eukaryotic cell. This cell, the primitive cell, is called basically prokaryotic cell. Karyon means kernel or seed, right? So this is what? Prokaryotic cell. This is prokaryotic cell. So prokaryotic cell is typically what? It is a primitive cell, and we are going to talk about this in this particular lecture. Let me tell you about this, right? Okay, this is the eukaryotic cell. This is prokaryotic cell which we want to discuss today. Pro means primitive basically, right? So let me tell you, and I hope you have got the basic idea what I want to talk here. Pro means simply prokaryotes, right? और यहाँ से क्वेश्चन आया हुआ था, ये बात आपको ध्यान रखना है, right? This is very important for you. Pro means primitive. Primitive means very old, ancient, right? It means English. It means ancient. It is not well developed. This is very not well developed, right? And what we are talking of? We are talking of karyon. What does karyon mean? Karyon simply means kernel, right? Kernel means seed, right? Karyon, it means nucleus. So typically in this particular cell, prokaryotes are basically, they are not well developed, very primitive cells, So there is no nuclear membrane. So definition is what? Nucleus is not well developed in these cells. This is what is prokaryotes basically, right? So you can see in this diagram there is some kind of membrane, and let me tell you, so here you have this region, it is called a nucleoid. This region is called a nucleoid. What is nucleoid? So it is written here also. It is called nucleoid. Nucleoid is what? It is aggregate of aggregates of DNA, right? What is DNA? DNA is a genetic material which has information, right? Genetic material which has information for next progeny, right? Right? They are free within membranes, right? They don't have. For example, cyanobacteria. Examples. If you want to talk about examples, and let me tell you something more about them. So, okay, let me tell you something more about them. They are examples are, for example, bacteria. You have bacteria like E. coli bacteria. For example, there are n numbers of bacteria. E. coli bacteria, right? Other example can be cyanobacteria, right? Cyanobacteria are bacteria, but sometimes they are also called as blue-green algae. Remember, they are not algae. They are not BGAR. Blue-green algae are basically bacteria. Remember this thing, okay? Right? Okay. So, if you were to talk about prokaryotes further, let me tell you, prokaryotes have typical size of 0.5 micrometer. So their size is, don't forget it, this size is around 0.5 to 1 micrometer. You can ask me what is 1 micrometer. This is typically read as micrometer, right? This is the symbol of micrometer. It simply means 1 part of a million. 1 part of a million, right? 1 part of a million. It is basically 1 micrometer is basically 10 raised to power minus 6 meter. So if you have 1 meter length and you break it into 1 million, right? One part will be called as 1 micrometer. And I have another slide for you. So let me show you this one to you. These are basic, So if you have a meter, they are penguin basically. These penguins are, their length is in meters, right? Meter, right? Meter is SI unit of standard unit, right? Of length, SI unit of length. Then there is millimeter. 1 meter has 1000 millimeter, and if you write it other way around, it has 10 raised to power minus 3 meter, right? It simply means if you break, 1 part of 1000, right? If you break 1000, if you break 1 meter into 1000 parts, then one part of 1000, is called millimeter, right? Then micrometer. 1 micrometer is 10 raised to power minus 6 meter. It simply means 10 raised to power minus 6 means you put 60 downstairs, 2, so ones, tens, thousand, 10,000, right? Lakhs, 10 lakhs, 10 raised to power 6, 2, right? So this is what? This is basically embryo, 8-cell embryo of human, 8-cell, this diagram is 8-cell embryo of human. Okay, this is in further nanometer is 10 raised to power minus 9 meter. So it is even lesser, right? So this is DNA, double helix structure of DNA, and this molecule is very small. So you should understand how small we are talking of, right? So we are talking about these small things. Let me tell you something more about these prokaryotes. Prokaryotes have ribosomes, right? तो इसमें एक क्या होगा, न्यूक्लियोइड होगा, डीएनए मटेरियल होगा, विच इज कॉल्ड न्यूक्लियोइड. इट इज नॉट पैकेज्ड अप, not packaged up like not packaged up as chromosomes. There are no chromosomes, it is basically naked. This material is naked, right? Not chromosomes, right? So you have ribosomes also there, ribosomes. This is nucleoid and this is a typical nucleoid. In this diagram you can see ribosomes are protein factories. They make up proteins for they are protein factory. They make proteins for the cell, right? Okay, quickly let me tell you a difference between then you have two things again. Let me write here cell. So cell has two types. One type is called prokaryotes, okay, other is called eukaryotes, right? One type is called prokaryotes, other type is called cell has two main branches. And first let me quickly discuss about prokaryotes only, right? Prokaryotes have again two branches. Detailed DNA analysis of these life forms, it was discovered that they have two different branches. One is called archaea, okay, one is called archaea bacteria, or otherwise simply archaea, right? It is archaea, ARCHAEA, right? Archaea. And others are basically eubacteria, or simply bacteria, or eubacteria. They are sometimes called bacteria or eubacteria. Okay, there are two branches. So I hope आपको समझ में आ रहा होगा. तो जो आर्किया है और बैक्टीरिया है, they have same difference as there is difference between prokaryotes and eukaryotes, right? Same difference is between archaea and bacteria, and bacteria is basically they look like bacteria but not bacteria, right? They look like bacteria. They look like bacteria. They look like bacteria, but they are not bacteria. Okay, just remember this thing. Another thing, they are they are sometimes called extremophiles. Some bacteria are also extremophiles, but archaeas are basically, they resemble ancient condition, when life was very difficult. So they are initial form, right? They live in harsh environment. Sometimes it resembles ancient life form, way back around 3.7 billion years ago, right? For example, they have different types. One type, for example, they are called halophiles. One example is halophiles. They are salt-loving halophiles. Okay, here we write halophiles mean loving. They are halophiles. Okay, one example is halophiles. What are halophiles? Halophiles are salt-loving archaea. What are they? They are salt-loving archaea, right? Salt-loving archaea, right? Another example, for example, let me write, they are sulfolobus. They can live, sulfolobus, right? Okay. These archaea can live about 90 degrees Celsius, right? Can live at 90 degrees Celsius temperature, right? And below pH less than 2, highly acidic environment like H2SO4, for example, sulfuric acid, right? Then therefore sulfur, H2SO4. Most interesting of these are methanogens, right? And they can ask you question about these methanogens. We are going to talk about methane, this is global greenhouse gas, and methanogens are basically, they are found in, for example, gobar gas plant or biogas plant, basically, where methanogens, they anaerobically digest glucose into methane also, right? They produce methane, methane, CH4, methane producing. What are they? They are methane producing organism, right? Bubbles, methane bubbles. Okay, all right, we'll talk about them. Then there are thermoacidophiles, right? Then there are thermoacidophiles. There are many examples. Thermoacidophiles, again, which loves both. Thermoacidophiles, they are like sulfolobus, right? So thermoacidophiles are, they again pH less than heat and acid loving, both. So they love both. ये कहाँ होते हैं? Heat and acid loving both. Acid loving both. These are archaea basically, right? So you have bacteria. Bacteria are typically, if you want to talk about bacteria, so a typical bacteria has like this kind of structure. It has outer cell wall structure, right? It has this cell wall structure, and we are going to talk about it later on. Cell wall structure. Then there is one region, where nucleoid. This is called a nucleoid. This region is called DNA, aggregates of nucleoids again. And you have, there are no specific cell organelles like mitochondria and all. These are protein factories. They are what? They are basically ribosomes. Okay, ribosomes are protein factories. And you have n number of bacteria, good bacteria, bad bacteria. For example, you have, E. coli bacteria, right? So you have many bacteria like E. coli bacteria. We have lactic acid bacteria. Lactic acid bacteria, they are useful bacteria. We have rhizobium bacteria, right? Rhizobium bacteria. Rhizobium bacteria has symbiotic relationship with, legumes. We have azotobacter bacteria. Azotobacter bacteria is basically free living nitrogen fixing bacteria, right? Then you have another bacteria. There are n number of bacteria. These are useful bacteria. Bacteria are basically decomposers also, right? You have, disease causing pathogenic bacteria, and famous one, for example, streptococcus bacteria, right? You have tuberculosis, mycobacterium tuberculosis bacteria, right? Tuberculosis bacteria, Yersinia pestis bacteria, for example, Yersinia pestis bacteria, which caused plague, plague causing bacteria. So we have n number of bacteria. Let's quickly talk about, this discussion is very long, but we are not going to keep it so long. Let's talk about eukaryotic cells. Eukaryotic cells is a well-developed cell, right? So you mean well-developed cell, right? Quickly we are going to talk about well-developed, carry on with nucleus. In this particular kind of cells, nucleus is well-developed, right? So quickly we are going to talk about this. Okay, so DNA is nicely packed up. I am writing somewhere. So this is nucleus. Nucleus has one more region. This is called a nucleus. Nucleus is basically, quickly, what is nucleus? Nucleus has, and nucleus, let me give you, we have another diagram also for eukaryotic cell. This is eukaryotic cell, full-fledged eukaryotic cell, right? So it is nuclear pore. Chromatin is basically what? DNA. It is mixture of DNA and proteins, specific proteins. These proteins are called histone proteins, and what they do actually, this is well-developed, cell. This cell is eukaryotic cell, right? So remember, this is eukaryotic cell, and eukaryotic cell is what? It is a very well-developed cell, right? Okay, well-developed cell, well-organized cell, well-developed cell. This is well-developed cell, right? Okay, so it has nucleolus also. These nucleolus are basically, they have RNA and proteins, right? And these RNA, for example, there are ribosomes, rRNA. So they are basically what? They make ribosomes. Ribosomes are made up of, for example, ribosomal RNA and proteins, right? There are transfer RNA and so on and so forth. These ribosomes are exported to cytoplasm. And let me again remind you all, this eukaryotic cell has three parts. Typically, it can have different shape, different size. Typically 3 parts simply means it has, this thing is called cell membrane, right? We are going to talk about it in detail some other class. It has cell membrane, right? This cell membrane is called plasma membrane. This is very important, and we have membranous cell organelles there. this one is called nucleus. So typically us eukaryotic cell has three distinct parts. One is called, the center part is called nucleus, which has nuclear pores, right? Then outermost part is plasma membrane, and then here it is called a cytoplasm. If we have animal cells has two more, you know, if we were to talk about plant cell has two more distinct two more part. It has cell wall also, and outermost part, and it has chloroplast. Chloroplast are cell organelles which are responsible for photosynthesis, right? So plant cells have these two extra parts apart from all of these things, right? Okay, so let me quickly discuss about this. Ribosomes, as I was talking about ribosomes. So ribosomes are what? They are protein factories. Ribosomes are protein factories, you know. Golgi apparatus, these vesicles are basically membranous bag. So there are some terminology. These Golgi apparatus are membranous bag, and they are responsible for membranes. They are responsible for making membranes, you know. And sometimes it is called, Golgi apparatus is called transport system. Golgi apparatus are called what? Transport system. What it does? Transport system of the cell, right? What it does basically, it, you know, modify, it packaged up and helps in transportation of the protein, and typically it makes membranes, and membranes are used so much, right? Then this nucleus we have talked about, it is called the brain of the cell. It is what? It is the brain of the cell. It is the site of cellular reproduction because DNA is genetic material. Then you have rough endoplasmic reticulum, RER, in short we call it RER. Rough endoplasmic reticulum has ribosomes attached, and wherever there are ribosomes are responsible for what? Proteins. Okay, just remember. And we are going to talk about in detail about all these things later on in other videos. Rough endoplasmic reticulum, let me tell you, it makes, you know, specific, highly specialized proteins which are typically transported to outermost part, you know, plasma membrane or extracellular fluid, right? Smooth endoplasmic reticulum, SER, they typically you know, produce fats, right? Okay, so we have peroxisomes, we have microtubules, lysosome, for example, lysosome is called suicidal bag, you know, it is called suicidal bag, but it is stomach of the cell. What it does? Stomach of the cell, right? Stomach of the cell, it means it digests worn out cell organelles, right? Then we have mitochondrion, most important structure, mitochondrion and chloroplast are supposed to be, they are supposed to be descendant of bacteria, free living bacteria. Mitochondria has their own DNA, you know, mitochondrial DNA, and it has its own ribosomes, right? And it helps in energy production. The function of this mitochondria is what? Energy production, right? So we will again talk about all of these things, right? Okay, so this video's content was basically about, let me quickly tell you, this video you know, there are cytoskeleton, something is missing, highly specialized protein, cytoskeleton, you know, they are network of proteins, you should know about them, network of protein. What they do actually, these network of proteins are basically responsible for physical architecture, you know, physical architecture of the cell, Okay, so we wind up this discussion today, not keeping this video too long, but this is very, very important topic, and we should know about the cell, because once we understand the functionality of the cell, we understand basic biological principles which can be applied to, as I have told you, interesting biological problems like cancer, for example, right? And we can talk about modern medicines. Today we wind up this discussion here. I hope you enjoyed this video, and if you really like this video, don't forget to hit like and share button. Till then, goodbye. Thank you so very much. Take care, guys. Thank you.